Good afternoon, everyone, and thank you for joining us for this first of 3 webinars that the BNA is running as part of our work to strengthen credibility in neuroscience, supported by the Gatsby Foundation. For this series of webinars, we've chosen to focus on reproducibility and robustness of in vitro neural models and what to consider in terms of best practices for these unique models. So this first webinar will feature Selina Wray from University College London, who will be telling us about reproducible research using stem cell derived neurons and organoids. So Selina will give a short 10-15 minute presentation, followed by time for questions. So if you do have a question, please type it into the questions tab, the little Q&A tab, so I can ask them at the end. The chat is for more general comments or issues. And I should also let you all know that this webinar will be recorded and it will be available afterwards on the BNA's YouTube channel for those who can't make it or wish to go back to the talk for any reason. So with that, Selina, I'll pass it over to you and thank you.
Thank you so much, Madeline and the BNA, for organising this series and for the invitation to speak. So over the next 15 minutes or so, I just wanted to talk a little bit about considerations when using induced pluripotent stem cell models for disease modelling. And the viewpoints that I will talk about today are largely based on our own experience, but also the questions that we commonly get asked when people come to us to collaborate or come to us because they're thinking about setting up these models. So this will be an introduction that I will just briefly kind of go over to say that the advent of patient-derived induced pluripotent stem cells has really transformed our approach to disease modeling, particularly in neuroscience. Now, we were fairly early adopters of this technology. We've been trying to use it for the past 11 years or so. And the reason I think this seminar series is so important is because quite often with new technologies, there's a tendency to really be a bit blind to their challenges or blind to their disadvantages and to kind of look at the system through kind of rose-tinted spectacles. And as this field has evolved, we've kind of become more open. There are more people involved in this discussion. And I think there are still a few areas that are necessary for us to reach a consensus agreement about, for us to really make sure we use this in the most robust and reproducible way. So these are questions that we have asked ourselves in the lab, and they're also questions that I'm commonly asked when people come to us for advice or for collaboration. And I hope I can touch on all of these today. So probably the one I will spend most time on is, should we use patient lines or isogenic controls that we can now make using CRISPR? What's the correct number of lines to use? Are we better to use many patients or many clones per line? Once we've got our induced pluripotent stem cells, what's the best way to make a neuron? How many times do we need to do that? And finally, and this is something that maybe we can discuss at the end, the majority of work at the moment is really focused on monogenic forms of disease. Can we apply these same modeling principles to understand the more common sporadic versions of the diseases that we are interested in? So our work is on Alzheimer's disease, and I'll kind of intersperse my thoughts with some of our data to show you how we've approached disease modeling in our lab. And we started off when only patient cells were really available. And that was our motivation for getting into this technology, because we were fortunate where I'm based at UCL to have some really kind of deeply genotyped and deeply phenotyped cohort. But of course, one of the biggest challenges that we became aware of in the IPS world very early on is the amount of variability that exists between different IPS lines. And I've highlighted here 2 of my kind of papers that have really influenced our thinking. And I know that Joe, who's organised this, has direct links to the papers that he will put alongside the recording. But what they show in these papers is that, first of all, the genetic background is the biggest source of variability. But also, when we think about whether we need more participants or we need more clones per participant, they really show it's better to use as many patients as possible. we always try to use a minimum of 3 patients and 3 controls, and actually this is the guidelines from Stem Cell Reports, which is the journal of ISSCR. So I think that's a good kind of minimum to bear in mind. Now, although the variability is a challenge, and you could perhaps argue that it's not necessary to deal with this variability anymore now that we can use isogenic lines, I still would like to make the case and kind of stick up for the idea of using patient lines in complement with isogenic lines. And the reason for that is we are always using lines which are from someone who has a phenotype of interest. There's a direct link to the disease. And also, I suspect that although this genetic variability is challenging, it can also be really important. We know that even in people with monogenic Alzheimer's, for example, there's a huge amount of clinical heterogeneity and variability in things like age of onset and disease duration. And we are now aware of several genetic modifiers that it will be important to use these models to kind of understand the impact of those. So this is just some data from our own work where what we can really see, I won't go into this in a lot of detail, the reference is here for anyone who is interested, but we're really looking at a very typical phenotypic readout for Alzheimer's disease linked mutations, where they shift the processing of these A beta peptides. And what we can see is that this measurement of the lengths of these peptides clearly separates the patients from the controls. So here we have a very robust phenotype, and we're able to be confident in this because we see it over at least 8 different individuals. So in spite of the genetic variability that exists, the phenotype is still present. However, there are still mutation-specific profiles. And again, I don't have time to go into the details, but there is information here that we wouldn't have captured unless we were using patient lines. And one of our interests is really trying to link this back to the clinical presentation of these people. However, we've also complemented this work using isogenic lines that we have made by Crispr, which really allow us to address questions that we couldn't ask using patient lines alone. So one of the things that's been a point of discussion in the field for kind of many years is whether certain mutations are gain or loss of function. Now, of course, this gives us a testable hypothesis because a loss of function would behave in a similar way to a gene knockout. And the use of CRISPR and the use of isogenic lines has really allowed us to test this in human neurons for the first time, because we can make genetic variants that wouldn't naturally occur within the population. And again, this is just showing that we were able to use one of our patient lines to make a wild type isogenic control, a homozygous version of the mutation that wouldn't naturally occur, and also a gene knockout for the presenilin-1 protein, which you can see here. And again, just going through this very quickly because it's published data, the citation is here. But this really gave us confidence in what we were seeing in our patient cells because we now have an allelic response to the mutation dosage on our phenotype of interest, which is A beta cleavage. But we can also see that the mutations behave differently to the knockout. So it's allowing us to kind of look at this question of loss versus gain of function. So this for me is really showing what we can achieve by using both our patient cells and our isogenic lines to complement one another. So this would be the summary of my kind of take-home thoughts, which is in an ideal world, we would use both to have the most robust insight into what's going on. There are, of course, advantages and disadvantages to both approaches, which hopefully I've outlined on the previous slide and I've also summarized here. But I would also acknowledge that there's a need for us to be pragmatic because I think as a field, you know, quite often the standard is set by labs who are doing IPS work full time, and we have access to the tools and resources to generate these large panels of cell lines. And I think one thing that we're starting to see in the field, which I think is really great, is resources which are enabling access to tools for the research community. And here are just a list of some different cell banks which provide iPS cells for neurodegeneration research. I would really recommend using cell banks where possible because they will have good SOPs for cell banking, and they will also provide basic quality control data on the cell lines, which I think, again, is really, it's good to know you're starting with something that's of high quality before you kind of get deep into analysis. I'd also highlight this recent initiative from the NIH that has just been announced, where rather than patient lines, they're focusing on using CRISPR to make over 100 different mutations associated with different forms of neurodegenerative disease within the same control line. And again, these will be widely available to the research community and help us to kind of do the best quality research and enhance reproducibility using these tools. So for my last few minutes, I just wanted to look at a different question, which is probably the second most common question that I'm asked, which is, okay, I've got my iPS cells. What's the best way to make a neuron? And the short answer is there isn't a best way. There are different ways that you can take stem cells and use them to make neurons. And instead of thinking, well, which is the best way, we need to reframe this as what's the most appropriate model to address the question that I'm interested in. There are different approaches such as directed differentiation, so really using small molecules to mimic development, permissive differentiation, which really harnessing the cell's ability to self-direct into certain cell types. And then finally, we're not really using these two methods, but I'm happy to talk about them in the discussion. There is transdifferentiation, which is the direct conversion of skin cells to neurons, and also induced neurons, so the overexpression of pro-neural transcription factors within iPS cells. But I'll just spend 2 or 3 slides to talk about some of the work we've done with these models. Now, for a long time, we were using 2D cortical neurons, but we've recently moved more into cerebral organoids. And the reason for that is because we have a couple of research questions where we believe a 3D system is more appropriate and more necessary. But one of the things that we've had to consider is actually our work is not single cell. We're looking at bulk population biochemical readouts. And what we can see here is comparing some of the data we have on 3 different control lines with 3 inductions of each line in both 2D and 3D. And we can see that what we were finding in 3D was actually quite a bit more variability in the levels of our protein of interest. And the reason for that is because in the organoids, they're really undergoing this permissive differentiation. So we're not giving them small molecule inhibitors in the same way that we do in 2D. And that leads to some variability in neuronal content, which when we're looking at the bulk population level, like we are on Western blot, has proved somewhat challenging for us. And so here's where I think it's kind of really important, even as somebody who is primarily interested in disease, to keep an eye out on what's happening in the world of developmental biology. And I hope Madeline won't mind me using this as an example, that we really have followed her work and seen how this protocol has evolved to become robust enough so that we can use it for the bulk population analysis that we use in my lab. And this is a paper where in Madeline's group, they've combined the organoid protocol with some scaffolding to, it's a very simple tweak that then gives the cells a more elongated structure. It means they have more exposure to the media, and this really means that there's a lot less organoid and batch-to-batch variability. And when Chris in my lab switched to this protocol and compared it with the previous work we were doing, we can see that all of a sudden we have a much more robust and reproducible batch-to-batch readout where we have really consistent levels of our protein of interest. And so that for us is an example of where we've really kind of followed the what's happening in developmental biology and in the organoid world, and been able to adapt that to make our disease models as strong as possible. And so I just have some final practical thoughts, if people are looking for tips to take away to the lab with them. The thing that I tell everyone is that quality control is it's not the thing we all get excited about, but it's really important. When you get new stem cell lines to your lab, I would really take the time to check that the karyotype is good, check that they're expressing pluripotency markers. The same is true of your differentiations. Every time we make neurons and every time we make organoids, some of them are reserved to make sure that we've got the cell type that we think we should have. Because just because we had it once doesn't mean we will always get that. So we need to do these checks every time. Do make sure that the lines have the mutations that you think they have. If you're doing disease modeling, we mycoplasma test every 2 weeks. We're quite often keeping these neurons for months and months at a time in continuous culture. So we need that repetitive testing, and really spend some time early on to build stocks of your cells where you know the karyotype is good. Build a big bank of cells at that early stage, so you know that you are always working with the highest quality material. Some other general tips, especially at the minute, I would try to bulk buy reagents. We're having some ongoing supply issues that we think are kind of post-pandemic related, where we've suddenly had not been able to get the reagents that we need. But I think this is good practice in any case to kind of have big batches, so you're always working within the same batch number of a particular reagent where possible. Not really talk too much about our phenotypes, but the approach we take, as well as trying to make the cells themselves as consistent as possible, is to use as many approaches as we can to identify a phenotype. So for example, we work a lot on tau splicing, and we look at this at the protein level with Western blot, with qPCR, with mass spec, with ELISA. We kind of take as many approaches as we can to really be certain that that phenotype is true. And of course, the work that I've shown you today is really looking at the direct readout of a mutation. As you move further away from the particular mutation, it gets more difficult. So I think having this direct readout is really important. So you at least have some indication that your model is kind of recapitulating something that's really linked to the clinical phenotype. So I'll stop there. I just wanted to highlight one reference that came out in Neuron about a year ago. Kind of captures a lot of what I've said today, and I think it's really useful in terms of moving towards best practice. So I would really recommend this paper. And also I would just like to thank my lab and my colleagues and collaborators who've really, we work together to kind of make our work as robust as we can, and I wouldn't be able to do it without their help. So thank you all for listening.
Thank you, Selina. That was fantastic. A really great start to this series. I think you brought up quite a few really important points that that really deserve probably more attention in this field, I think. So yeah, so we have time for questions. So if any of our viewers do have a question, feel free to type it in the Q&A tab. There's one question so far from Katja Salbaum. Hi, thanks for this great talk. Your data suggests that presenilin 1 mutations are a gain of function mutation. Is the fact that the mutation acts differently than a knockout sufficient to conclude that all presenilin mutations are gain of function mutations?
That's a really good question. And I should say that because of time, I kind of only showed you one snapshot of the data that we have around here. So we've also combined, so I agree with the comment that it's not enough on its own to show that it's loss of function. We've looked at multiple readouts based on the enzymatic activity and also on other functions of the presenilin protein. And so, for example, as well as being a kind of a component of gamma secretase, we know that presenilin can act as a molecular chaperone. And what we see with our mutations is they only affect the enzymatic activity, they don't affect the chaperone activity, whereas obviously the loss of function affects all functions of presenilin. In addition to that, we've also used small molecule inhibitors so that we can complement the genetic approaches with alternative approaches to really be confident that we see the same thing, looking from multiple angles. But I think there's another important point there, which is, it's not possible for us to make isogenic controls and knockout on every background that we use. And we have to accept that as a limitation of what we're doing. And so we're drawing the best conclusions we can based on kind of multiple approaches and the limited lines that we have.
And so there's sort of a second part to that, but that actually kind of goes into something that I wanted to ask you as well, which is kind of leads on from what you've just said, which is, there's these different approaches, the patient cell lines that you've shown so nicely, and then you've gone back and repaired the mutation. So you've got that kind of an isogenic control. But then you also showed the initiative from the NIH where they're taking a control cell line and then introducing the mutation. So they're both isogenics, isogenic comparisons, but maybe you could speak to the sort of benefit of each of those and what are the strengths and advantages of each of those different kinds of approaches?
Absolutely. So I think with the NIH initiative, the advantages are that they are using a parental line, so the control line that they're using as the basis for their gene editing is really well characterized, both in terms of quality control of the line, differentiation potential of the line, and also the genetic background of the line. So for example, we know that this particular line is neutral in terms of Alzheimer's risk genes. So risk variants that have been identified through genome-wide association studies, for example, it doesn't have a high risk score for Alzheimer's disease. What they're also doing in this initiative, which I really like, is that they are making the causative mutation and then they're also reverting that back to wild type. So you've kind of got the control at either side of the process, if you like, which I think is really powerful. For me, though, the advantage of using a patient line is I know for sure that that person had the disease that I'm trying to understand. And I think that's really powerful when we're trying to understand clinical heterogeneity. So for example, within a family of people who all have the same presenilin mutation, why is it that the age of onset can vary by 10 years? And I think it's highly likely that at least some of that is down to other variability in the genome that we can then use these cells to maybe understand. And we're also doing a lot of comparative analysis with cells, CSF, and postmortem brain tissue from within these same patient groups. So I think the two approaches are highly complementary, but I definitely, I would say that isogenic lines, as great as they are, as much as we're using them ourselves, are not going to replace patient material just yet.
Great. So there is another question. I think this is a really fantastic question from Irene Luca. She says, hello, thanks for the interesting talk. I was wondering what your thoughts are on using IPSC derived neurons for developing therapies, and should the same level of scrutinization of the effects of line and other sources of variability be looked at when performing those types of approaches?
Yeah, this is a really good question, a difficult one to answer. I mean, I think obviously when people are looking at developing IPS to use in cell therapy, the level of quality control that needs to be done is in a different world from what we're doing in the lab in terms of making sure they're GMP grade and really, when I talk about looking at karyotypes, I'm really looking whether we've lost or gained a chunk of a chromosome, whereas here it needs to be much more kind of fine-tuned. In terms of the level of, sorry, just remind me of the question again. It was the level of scrutinization.
Yeah, the level of scrutiny, sort of some of the QC that you talked about, for example, does that need to be a higher level when you're talking?
Yeah, no, I absolutely think it does. And of course, there's also I don't feel fully qualified to speak to this because we're not going down a therapeutic route ourselves, but there's also a lot of considerations around kind of matching donor background to the recipient to kind of avoid or minimize chances of rejection as well. And I think, one of the things that I often think about is how we're using oncogenes to generate these IPS cells, and we have to do whatever we can to make sure that any cells that are being used in therapy don't have the potential for kind of to become cancerous. So yeah, I think it's absolutely in a different world to what we're doing in the lab.
Yeah. So maybe if I have time, maybe I'll ask you one last question. You're working on neurodegeneration. So that's obviously a quite late onset disorder or class of disorders. So I wonder if you could speak about how you sort of view these kinds of in vitro approaches, 2D or 3D, all of which are coming from these pluripotent stem cells and are therefore, very sort of developmental, and what that means for these studies of more late onset disorders.
It's a really good question. I think something that is sometimes we don't talk about it in the field enough, actually, that we are using developmental system to kind of approach these diseases of aging. And there's some really nice work that has been published recently from the Pasca lab on organoids, where they've done single cell sequencing throughout development into these kind of really, I'm saying old cultures, 600 days. That's really old in terms of culture, and of course in terms of development and aging it isn't. But what they have shown is that they really kind of faithfully play out this developmental clock and undergo pre to postnatal transitions in, for example, the splicing, switching of NMDA receptor subunits. We've got our own data which complements that, where we've looked at tau splicing in development. And again, we're seeing that we're not getting the adult tau splicing until these cells are very, very old. So that is a disadvantage that we need to be aware of. However, it's clear that when we've got a disease-causing mutation, phenotypes are there. So I almost think, I view it as an opportunity to understand why are we able to cope with these mutations for so long? What is making these early cells resistant to actually degenerating until we're much older? So although it is a challenge, I think there's an opportunity there to really understand the early stages of disease, and also maybe to gain insights into what could be protective if we know that these fetal cells are not succumbing to that pathology. There are also, of course, some alternative approaches such as transdifferentiation, and there's some nice work suggesting that if you directly convert a fibroblast into a neuron rather than going via IPS, it will retain its epigenetic markup that signifies how old the donor was. So I think there are some practical approaches as well to maybe incorporate age into this system.
Mm-hmm. So there is one more question, and we do have 3 more minutes, so maybe I can ask it, or at least sort of frame it in a similar kind of vein that I also wanted to go, actually, which is when you're comparing these patient cell lines, there is quite a bit of variation even between control lines. And so when you're comparing these different cell lines, we've talked about isogenic comparisons, but comparisons between, like you mentioned, 3 controls and 3 disease lines, for example. Do we need to also think about sort of also, have you thought, or are you thinking about the relationship between those donors? So you mentioned there's the genetic basis for a lot of that. So are you using, or do you think this is something we should start doing, controls that are actually related, like familial members,
I think that's a great idea. I think where possible in our lines, when we ask people who participate in our research, we try to take unaffected siblings where we can. It's not always been possible for us to do that, in which case we try to use age and sex match controls. In a way, so I think reducing the variability by using related donors is a great idea, but in a way, if you can see phenotypes above the variability from people who are not related at all, I think that even gives us more confidence that those phenotypes are real. So again, I think it's kind of good to have both approaches, but definitely if there's the option to kind of use people who are related, then it's not isogenic, but it's the next best thing, I think.
Okay, well, I think that brings us to the end. Thank you very much, Selina. Thank you to all of the attendees. I hope that this was helpful. And the next one of these webinars will take place at the same time, same place next week. I hope that you can join us for that one. Thank you.